Welcome to Mythos AI, your source for Cliff's Notes on complex subjects. Join Anna and Phil for yet another engaging conversation.
Welcome to the deep dive. Today we're looking into something pretty fascinating, a new AI model from Google DeepMind. It's called AlphaGenome. And our goal here is to explore this area known as the dark genome. That's the 98% of our DNA that doesn't code for proteins. It's been a huge mystery for a long time. We want to understand why it was such an enigma, how AlphaGenome is helping to, well, crack its code, and what this all could mean for medicine and, future discoveries.
Right. AlphaGenome is essentially a foundational AI. Think of it as an interpreter for that non-coding DNA. Its main job is to figure out the causal links, the cause and effect between small genetic changes, variants, and what actually happens biologically. And it's built to handle really long stretches of DNA sequence, up to 1 million base pairs, and make predictions about quite a few different biological activities. RNA splicing is a key one.
Okay, let's back up a bit. For a long time, scientists called this stuff junk DNA. That sounds pretty dismissive. Why that term? What did people think it meant?
Yeah, junk DNA was a prevailing idea for quite a while. It came partly from the focus on protein-coding genes. They seem like the only important parts. There was also this early population genetics idea, suggesting that if too much of our DNA was essential, the natural rate of mutations would just be devastating. Too many critical hits, basically. So the implication was that most of our DNA was just, well, evolutionary leftovers, no real job.
But that turned out to be wrong.
Very wrong, yes. Big projects like the ENCODE project came along later and systematically mapped activity across the genome. What they found was striking. At least 80% of the genome is biochemically active. It's not junk. It's more like a really complex regulatory control system. And crucially, when you look at genetic variants linked to common diseases, things like diabetes or heart disease, found in these large studies called GWAS, over 90% of those variants fall within these non-coding regions.
Okay, a regulatory control system. That sounds important. Can you unpack that a bit? How does this non-coding DNA actually control or conduct our genes? What are the key parts?
It's a great analogy, actually, the conductor. Imagine the genes are the instruments in an orchestra. The non-coding DNA is both the conductor and the sheet music. It tells each gene, each instrument, when to play, where, how loudly. That's how we get all the different types of cells, liver cells, neurons, skin cells, all from the same set of genes.
So what are the specific elements, the notes on the sheet music, maybe?
Well, there are several key types. You have promoters. These are right next to a gene, like an on-off switch. They're binding sites for the basic machinery that starts reading the gene. Then you have enhancers. These are more like volume knobs. They can be really far away from the gene, thousands, even millions of base pairs away.
Millions. How do they work from that far?
The DNA actually folds up in 3 dimensions inside the cell nucleus. So an enhancer that's far away linearly can physically loop over and touch the promoter of its target gene, boosting its activity. And conversely, you have silencers. These act like mute buttons, binding proteins that shut down gene activity. The specific combination of these active elements, which enhancers are on, which silencers are active, is what defines a cell type and its function. And a change, a variant, in one of these elements might only matter in certain cells.
That sounds incredibly intricate.
Yeah.
So what made it so hard for scientists to figure this all out using older methods? Why the dark genome label?
It really was a wall of complexity, you could say. First, just the scale and the distance issue we mentioned. Trying to link an enhancer millions of bases away to its gene experimentally is tough. And in those GWAS studies, you might find a region linked to a disease, but it could contain hundreds of variants. Pinpointing the one causal variant is like finding a needle in a haystack.
Okay, scale is one thing. What else?
The other big challenge is context dependence. An enhancer might be crucial for a neuron during brain development, but completely inactive in an adult liver cell. So to really understand a variant, you'd ideally need to test it in, hundreds of different cell types and conditions. And experimentally, that's just not feasible at a large scale. Computationally, the models before AlphaGenome had this trade-off. They could look at long DNA sequences, or they could predict things at very fine single-base detail, but they couldn't really do both well at the same time.
Right. The scale versus resolution problem. So how does AlphaGenome manage to do both? What's special about its architecture?
AlphaGenome uses a hybrid approach, combining 2 types of neural networks. It takes in a long sequence up to 1 million base pairs, which is enough to capture most of those long-range interactions. But critically, it also outputs predictions at single base pair resolution. This means it can model the effect of changing just one single letter of DNA, a SNP.
How does it manage that combination?
It has two main parts. First, convolutional layers, which are good at spotting short, recurring patterns in the DNA, like specific sequences where proteins bind. Then that information feeds into a transformer core. This uses the same kind of attention mechanism you find in large language models like ChatGPT. This allows the model to weigh the importance of different parts of that million base pair sequence and understand the complex, long-range dependencies, essentially, learning the grammar of how regulatory elements talk to each other across distances.
That's clever. So what kinds of things can it actually predict? You mentioned RNA splicing earlier. Why is that particularly significant?
It's designed as a single, unified model that predicts a wide range of things. It's trained on huge amounts of public data from projects like ENCODE. In total, it predicts over 5,900 different signals for the human genome and over 1,100 for the mouse genome across 11 broad categories. These include things like how active a gene is likely to be, gene expression, where the DNA is physically accessible, chromatin accessibility, where specific regulatory proteins are binding, transcription factor binding, and even how the DNA is folded in 3D space.
And the splicing part.
Right, the RNA splicing prediction is a really important advance. Splicing is the process where the initial RNA copy of a gene is edited bits are cut out and the remaining bits are stitched together. AlphaGenome doesn't just predict if splicing happens, but precisely where the cuts are made, how often different versions are produced, and exactly which pieces are joined together. This is huge because mistakes in splicing caused by genetic mutations are known to cause many severe genetic diseases, things like spinal muscular atrophy or some types of cystic fibrosis. Understanding these errors at a predictive level is a big step forward.
That really does sound like a leap.
Yeah.
How accurate are these predictions, though? What did the performance tests show?
The evaluations were pretty comprehensive. AlphaGenome was compared against the best existing models across a range of tasks. For predicting general genomic activity, like where proteins bind it, outperformed the previous best models in 22 out of 24 different tests. And for predicting the functional effect of a genetic variant, it matched or beat the top models in 24 out of 26 evaluations.
So it's holding its own, even against specialized models.
Yes, and often exceeding them. It consistently did better than models designed only for splicing, like Splice AI, or models designed only for chromatin analysis. For example, predicting how a variant changes gene expression levels, it showed about a 25% relative improvement over the previous leading model, Borzoi. That suggests it's learned some genuinely fundamental rules about gene regulation.
Impressive results. But no technology is perfect. What are the known limitations or constraints of AlphaGenome right now?
There are definitely limitations to be aware of. Firstly, access. It's currently only available for non-commercial research, and that's through an API with usage limits. This means doing very large-scale analyses, like across entire genomes from many samples, isn't really feasible for external researchers right now, and the actual model weights haven't been released publicly.
So it's powerful, but access is controlled. Anything else?
Yes, it's important to stress it hasn't been developed or validated for clinical use diagnosing patients, for instance. Also, while it handles long sequences, its predictive accuracy does start to drop off for regulatory elements that are very far away, say beyond 100,000 base pairs from the gene, even within that million base pair window. And finally, it's modeling the DNA sequence and its direct consequences. It can't currently account for more complex biological layers like the timing of events during development or the influence of environmental factors on gene activity.
Okay, that makes sense. You refer to it as a predictive oracle. How does it actually function like that for a researcher? What does doing an in silico experiment look like, and how does it change things?
The oracle function really comes down to variant effect prediction, or VEP. It's incredibly fast. A researcher basically gives the model two sequences, the normal or reference DNA sequence for a region, and the same sequence but with the specific mutation of variant. AlphaGenome then predicts all its outputs, gene expressions, splicing, etc. for both sequences. It then calculates the difference, what changed because of this one mutation.
Yeah.
And this whole process, this computational experiment, takes less than a second on a powerful GPU.
Less than a second compared to lab work.
Exactly. Compared to potentially months or even years of complex, expensive lab experiments trying to figure out the same thing, it dramatically shifts the bottleneck. Generating a plausible, data-driven hypothesis about what a variant does becomes almost instantaneous and virtually free. The effort then shifts to validating the most promising predictions in the lab. This is especially powerful for studying rare variants, the kind often involved in severe inherited diseases, which are very hard to study traditionally.
Can you give us some concrete examples where AlphaGenome has shown it can predict mechanisms we already knew were involved in diseases, sort of like a reality check.
Yes, they've published several examples demonstrating this. One involves a type of cancer called T-cell, acute lymphoblastic leukemia, or TAL. There are known non-coding mutations near a cancer-driving gene called TAL1. AlphaGenome was able to analyze these mutations, and it correctly predicted that these mutations create a new binding site for a different protein, a transcription factor called MYB. This effectively hijacks the regulation, causing TAL1 to be turned on inappropriately, which drives the cancer. That matches what was found experimentally.
Okay, that's a strong confirmation for cancer.
Yeah.
What about other areas?
Well, with its strength in splicing prediction, it's very relevant for diseases caused by splicing errors. It can model how a single DNA change might cause a crucial part of a gene, an exon, to be skipped during RNA processing, or cause an incorrect cryptic splice site to be used instead. Both can lead to faulty proteins. This helps researchers make sense of variants found in patients with conditions like spinal muscular atrophy or cystic fibrosis much more quickly, and it's also proving useful for interpreting those Genomics results we talked about. It can take a variant statistically linked to a disease and predict what it likely does functionally and whether it increases or decreases gene activity, providing concrete hypotheses to test.
That really clarifies its utility. But how definitive are these predictions? If AlphaGenome says a variant causes X, is that the final answer?
That's a crucial point. The outputs are probabilistic hypotheses, not absolute certainties. Think of it more like a sophisticated weather forecast than a mathematical proof. It gives you a likelihood, a score, suggesting that a variant might alter gene expression by a certain amount or is likely to disrupt splicing. It doesn't give a simple yes or no.
So they guide experiments rather than replace them.
Precisely. They accelerate science by telling researchers, based on what we know about genomic grammar, this variant is the most likely culprit, and this is probably how it's causing problems. You should test this first. It dramatically increases the efficiency of the experimental process, but it's vital to remember it can't predict complex outcomes like, will this person get diabetes? That involves way more than just one variant's effect, multiple genes, environment, lifestyle, things beyond the model's current scope.
Got it. Now, AlphaGenome isn't working in isolation. It's mentioned as part of a larger AI stack. How does it connect with other AI tools to create a kind of workflow?
Yeah, this is where it gets really interesting. The vision is to connect different AI models to mirror the flow of information in biology, what's often called the central dogma. RNA makes protein. So the workflow looks something like this. First, you have AlphaGenome, DNA to RNA. You input a DNA variant, and it predicts the impact on gene expression and, crucially, the resulting sequence of the messenger RNA, mRNA, including any splicing changes.
Okay, so you get the predicted RNA sequence. Then what?
Then that predicted mRNA sequence is computationally translated, RNA to protein sequence, into the sequence of amino acids that make up the corresponding protein. And finally, that amino acid sequence is fed into AlphaFold 3, protein sequence to 3D structure. AlphaFold 3 predicts the complex three-dimensional shape of that protein and potentially how it interacts with other molecules like DNA, RNA, or even drugs.
Wow. So you can go computationally from a single letter change in the non-coding DNA all the way to a 3D picture of the potentially faulty protein it produces.
Exactly. It's a way to computationally model the entire cascade of effects, from the genome to the functional molecules of the cell. It aims to make biological discovery much more systematic, almost like an engineering process.
And where does Isomorphic Labs, the Alphabet company, fit into this picture, especially regarding drug discovery?
Isomorphic Labs was essentially set up to apply this AI stack, this integrated workflow, directly to the challenges of drug discovery. They call it an AI-first approach. By modeling the precise molecular consequence of a disease-linked variant, understanding exactly how it changes the protein's 3D structure or function, they aim to identify better drug targets.
How does the 3D structure help?
Knowing the detailed 3D shape of a malfunctioning protein is incredibly valuable for designing drugs. You can computationally search for, or even design from scratch, small molecules that might bind to that protein. The goal is to find molecules that could correct the protein's function, maybe stabilize it if it's misfolded, or block its activity if it's overactive. This AI-driven approach promises to make the very early stages of drug discovery faster and hopefully more successful.
It's a compelling vision. What's next for this AI biology stack? Are they planning to add more layers or integrations?
The potential for extension is definitely there. You could imagine future models incorporating even more layers of biological information, for instance, epigenetic modifications like DNA methylation, which also influence gene activity. The grand vision, which leaders at DeepMind and Isomorphic have talked about, is moving towards a virtual cell. a comprehensive computer simulation where you could perturb one component, introduce a mutation, add a drug, and accurately predict all the downstream effects throughout the cell.
A complete simulation of a cell. That's ambitious.
It is, but it shows the direction. And there's also a really powerful synergy with experimental tools like CRISPR gene editing. AI predictions can guide the design of very precise CRISPR experiments to quickly test hypotheses in the lab, creating this rapid feedback loop between computation and experimentation.
This really does sound like a fundamental change in how biology research is done, shifting the focus from the lab bench to the computer, at least initially.
It really does feel like a paradigm shift. Traditionally, research was often hypothesis limited. You had an idea, then you spent a long time testing it in the lab. Foundational models like AlphaGenome act as incredibly powerful hypothesis generators. They can sift through complexity and propose thousands of plausible, testable ideas automatically.
So the scientist's role changes.
It evolves, yes. Less about coming up with the initial hypothesis from scratch, perhaps, and more about becoming a skilled curator, interpreter, and validator of the hypotheses the AI suggests. You query the AI, get prioritized predictions, design target experiments. It could dramatically compress the timeline of discovery, turning years of work into potentially months or weeks for certain kinds of problems.
What could this speed up mean directly for patients, for precision medicine?
The potential impact is huge. Think about diagnosing rare genetic diseases. Today, it can be a long, painful process, a diagnostic odyssey. In the future, you can imagine sequencing a patient's genome, feeding their unique variants into a model like AlphaGenome, and getting back a ranked list of the most likely disease-causing mutations and exactly how they're predicted to disrupt cellular function. It shifts diagnosis from elimination to prediction.
And for treatment.
If you understand the precise molecular problem caused by a specific mutation in that patient, you can start thinking about treatments tailored to fix that specific problem. It moves medicine closer to being a true engineering discipline, targeting the individual's unique biology.
The potential benefits are clearly enormous. But technology this powerful often comes with complex ethical questions. What are some of the concerns being raised about access, control over data, and this concentration of power?
These are really important discussions that are happening now. One major point is access. As we mentioned, AlphaGenome isn't open source, it's proprietary, and access for the wider research world is currently restricted. This naturally creates a gap, a kind of two-tiered system where the developers and their affiliates have a significant advantage over academic or nonprofit researchers. That's led to some criticism.
And what about the data it was trained on?
That's the second point, the commodification of public data. These models are trained on vast data sets like ENCODE that were generated through massive public funding over many years. So while the raw data is technically public, the cutting-edge tool needed to really unlock its value is now proprietary. It raises questions about whether the value derived from public investment is being fairly shared. It echoes some debates from the original Human Genome Project era.
Access, data.
Yeah.
What else?
The third big area is corporate gatekeeping and the concentration of power. When one entity controls such a powerful tool for interpreting the fundamental code of life, they gain immense influence. This raises questions like, who gets to decide which diseases or biological questions get prioritized for research using these tools? How might commercial interests shape the direction of science? How do we ensure the benefits reach everyone equitably, not just those who can afford it? There's a risk of becoming epistemically dependent on a private company for fundamental biological insights.
It's a fascinating duality.
Mm-hmm.
On one hand, AlphaGenome is this incredible tool lighting up the dark corners of our genome, potentially revolutionizing how we understand and treat disease, almost turning biology into engineering.
Exactly. It offers immense promise, but it's also, as you say, a double-edged sword. The very power that makes it so exciting also brings these significant ethical and societal challenges about control, access, and equity that we really need to grapple with.
We seem to be entering an era where we're moving beyond just observing biology to being able to simulate it with increasing accuracy. Maybe the big question for all of us then is, how do we navigate this? How do we ensure these powerful new capabilities are used wisely and truly for the benefit of everyone?
Thanks for listening to Mythos AI. Stay tuned for more concise insights. And remember to hit the like button and subscribe for future episodes.